good afternoon to Canada. Welcome to the second in the series of lectures at the Society for Molecular Biology. Student section is organizing to mark the worldwide event of World Antimicrobial Awareness Week. Today or tonight with us, we have Professor Laurie L. Burrows from McMaster's University in Ontario, Canada. She is a full professor of microbiology and a well-renowned expert on the structure of type 4 pillars. She knows everything about its structure, function, and its regulation. Her laboratory also studies the phenomenon of biofilm formation in Pseudomonas aeruginosa, and this is going to be the basis for her talk today. She's going to teach us how we can use biofilm knowledge in development of new drugs. So, Professor, good day to you again. We are very happy, and we can start whenever you are ready. Thank you.
Great. Thank you so much for the invitation. I really appreciate it. And I'm glad to be able to have the chance to speak to you on World Antibiotic Awareness Week. So I'll start my slides. Can you just let me know if you can see them?
Yes, we can see them very well.
Okay, great. Thanks. Okay, so I'm going to talk to you today about bacterial biofilms, what they are and how we use them to find new antibiotics. So that is a little bit backwards. Normally people are looking for antibiotics to kill biofilms, but we have actually trained our biofilms to help us look for antibiotics. So just a quick disclosure statement, some of the work that I'm going to talk about, there's a provisional patent on that chelator combination. So the outline of my talk today, I'm going to tell you a little bit about what biofilms are and why and how they form. I'm going to tell you how biofilms tolerate antibiotic treatment and how this can enable the development of resistance to antibiotics, which, as we know, is a huge and growing problem around the globe. And last, how we can take advantage of bacterial biofilm behavior to find new antibiotics. So the traditional definition of what a biofilm is communities of bacteria attached to a surface. And I put "and other organisms" because usually bacteria are not the only organisms in a biofilm, unless we're studying them in the lab and we've made an artificial, very monospecies biofilm. So in this picture that I'm showing you of, the forest, the stream, the rocks, you would find the biofilms on the edges of the rocks, mostly near the water line. So these are very thick biofilms. But all of the surfaces in this picture have bacteria biofilms on them. It's just that some places it's more obvious to the naked eye than others. You find other organisms where bacteria are because they go there to eat the bacteria. So that's the basic definition of a biofilm. In terms of human medicine, which is where we usually study biofilms, at least in my lab, biofilms are typically associated with medical devices. So it's been estimated that somewhere between 65% and 85% of human infections are actually biofilm related. And this is from an older paper now, but I really love this diagram because it shows you all of the many surfaces that bacteria can grow on in your body. So we have examples everywhere from sinusitis, which is, the sinuses in your head. This is very, very common biofilm infection. Ventilator-associated pneumonia. We've seen this during COVID, right? Because a lot of people are getting ventilated because they've had COVID and now they're getting secondary bacterial infections. So pretty much any medical device that you can think of can be contaminated by bacteria. And this is a problem because, if it's something as simple as a urinary catheter or a urinary stent and it gets contaminated with a biofilm, you can remove it. But if it's something more serious, like an implanted joint, so a hip or a knee, or even in the cystic fibrosis lung, you can't remove that thing easily, right? So it becomes very difficult to treat those types of infections. So one of the hallmarks of biofilms is that they produce their own matrix. And this is a mixture of sugars, proteins, lipids, and DNA that surround the bacteria. So I'm showing you two images here. The one on the left is a scanning electron micrograph of a Listeria monocytogenes biofilm growing on stainless steel. So Listeria is a foodborne pathogen. You get it if you eat unpasteurized cheese sometimes, or meat, like sliced meat, like ham that has been contaminated and then eaten directly without being cooked. And this organism can live in cold and high salt conditions, and it likes to form biofilms on food processing surfaces. So in this electron micrograph, you can see this clump of cells here, and they're surrounded by this goopy stuff, which you can't really see because in order to do this kind of microscopy, you have to dry the bacteria down. So you take all the water out of the matrix, and so you can't see it very well, but you can see the effect of what the matrix does. It basically clumps the cells into a big ball. And if you're trying to think about how you would treat an infection like this, you can imagine that treating single cells like the ones on the background stainless steel is pretty straightforward because the antibiotic can get to them, but it's a lot harder to treat bacteria that are growing in a big clump like this. So on the right-hand side, there is another micrograph. This is from a confocal scanning laser microscope. And the advantage of this type of microscopy is you can look at live cells while they're still hydrated. And so the light blue material that you see in the background of this picture is the matrix. So the bacteria are all held together loosely by this matrix that they have made themselves. So they make the sugars and release the DNA and the proteins and the lipids. And that basically surrounds them almost like a force field, right? So they make their own universe, kind of like in the actual Matrix movie, right? They live in an alternate universe. So this universe is protective to the bacteria. So this is a pretty standard picture of a biofilm life cycle that you would see in most review papers or textbooks if you read about biofilms. So we think of it as sort of a continuous cycle, and whether it starts with the left side or the right side doesn't really matter, but it's a continuum, right? So individual cells or free swimming cells we call planktonic, right? So like plankton in the ocean, they're free floating. Bacteria don't prefer to live this way. They prefer to live on surfaces. So when they come into contact with a surface, they will attach to that surface, and they will initially attach temporarily and they can leave again. But if they like what they find there in terms of it's a good environment, it's got food, there's no predators, they will become permanently attached. And once they do that, they will start to produce matrix around them. So that's what this gray material is. So this initial step is attachment, then microcolony formation. So micro meaning small. And eventually that will develop into what we call a macrocolony, which is this big clump of bacteria surrounded by matrix. This is not the end of the cycle though, because if this clump gets too large, bacteria can actually release themselves from the matrix because they'll start to make enzymes that break down the matrix so they can escape, right? Because you can imagine there are gradients of oxygen and food, and the guys in the middle of this biofilm will be getting less food and oxygen. So they can actually trigger a dispersal cycle and let themselves out of this biofilm. Or sometimes the biofilm gets so big that pieces of it will just break off, right? So you can imagine in that stream that I showed you, if the biofilm grows too large, the force of the water can break off pieces and they'll go downstream and stick somewhere else and start over again. So just a few words about, why do bacteria do this? And, it's not just bacteria, fungi also do this. So Candida, for example, which is a opportunistic yeast pathogen, will also make biofilm. So why do they do this? So one of the reasons is that nutrients concentrate on a surface, right? So in that stream that I showed you, there's more nutrients concentrated on those rocks than there are just floating in the water. So bacteria will be attracted to where there are higher levels of nutrients. Growing as a group, as a biofilm, promotes communication between bacteria, right? So bacteria don't have eyes or ears. They communicate with small molecules. They communicate chemically, and it's a little easier to send a chemical to the person that you're talking to if you're really close to them. If you're too spread apart, the chemical signals can diffuse away and the communication is lost. So this communication can activate group-specific behaviors, right? So the bacteria can, as a group, activate a behavior that will not happen if they're alone. They can transfer DNA fragments to one another, and even cell contents. They can actually fuse together and transfer cell content. So this transfer of DNA is important in terms of antibiotic resistance, because that DNA can contain elements of antibiotic resistance. The bacteria growing in a biofilm can share what's called public goods. So for example, this could be an enzyme that's secreted to break down a substrate that the bacteria are growing on. This could be a thing that benefits the same species, or could benefit other species growing together with the first one that is releasing this enzyme. But this phenomenon also leads to cheating. So bacteria can actually cheat. You can have the evolution of mutants that can use the results of the enzyme activity, but they don't actually make the enzyme. So they have a benefit because they're not making the enzyme, and so they're not wasting energy. So there's a whole subfield of biofilm research that is called Socio Microbiology. And one of the things they study is how bacteria control cheaters. So how do they prevent cheaters from taking over the population? It's a really interesting concept. And last, bacteria and fungi make biofilms because it will protect them. So it protects them from bacteriophages, which are viruses that kill bacteria. So having that matrix or that force field around you makes it harder for phages to find you and bind to you. It protects them from predators. So lots of other things eat bacteria. So, you know, all the way from protists to nematodes to larger organisms. If you're in the human body or the animal body, being in a biofilm can protect you from immune cells, right? So if you're growing as a big clump, like I showed you, it's harder to get taken up by a neutrophil or a macrophage because you're bigger than the neutrophil or macrophage. It protects the bacteria from desiccation, so drying out, right? When you're very small, drying out is a big problem. So if you lose water, you're going to die. So that matrix that I showed you is very hydrated and full of water. And last, it protects them against antibiotics and disinfectants. And I'm going to say more about that in a second. So when we look at this biofilm life cycle again, the planktonic cells are considered antibiotic susceptible. And this is how we test for susceptibility in a lab, right? So if you have an infection and you send it for culture and they're going to do a test to find out what antibiotics it's susceptible to, they will grow the bacteria as a broth culture, right? So this is how we are looking at bacteria in the lab, even though, as I told you, that's not actually how a lot of infections grow. So 65 to 85% grow as biofilms. So, you know, we have to question whether this is the right way that we should be testing bacteria for antibiotic susceptibility. In the biofilm macrocolony, the bacteria are considered antibiotic tolerant. So I want to make this distinction between tolerance and resistance. So resistance is a permanent thing. So if you become antibiotic resistant, that's an inheritable trait, right? So you're going to pass that trait to your progeny. Antibiotic tolerance is more about the way you are growing, because if you take this biofilm and you disaggregate it, so you disperse all the cells back into a planktonic form, they will become susceptible again. So they're only tolerant of the antibiotic while they're growing as a biofilm. They haven't had any permanent changes within that biofilm that make them forever resistant, right? So then we just think of that as tolerance. So it's something about the way they're growing. So the things that contribute to tolerance of antibiotics when bacteria are growing as a biofilm is, first of all, the limited ability of antibiotics to penetrate into this big clump, right? So the matrix, because it's got sugars and DNA, is often highly charged, and many antibiotics have charges on them as well. So a typical example is aminoglycosides. They are positively charged antibiotics. And if you have a lot of DNA and negatively charged sugars in the matrix, those antibiotics cannot diffuse easily because you're going to have charge interactions. And essentially, they're going to get stuck in the outside of this shell and not reach the bacteria inside. Inside this big clump, as I mentioned earlier, you're going to have some differences in physiology because some bacteria are going to have different access to nutrients and oxygen than others. And many of our antibiotics that we use today target processes like cell wall synthesis, DNA synthesis, RNA synthesis. And if you're not actively growing because you're starving for food or oxygen, those antibiotics have less effect on you. So essentially what that means is rapidly growing bacteria within this biofilm will be more susceptible to the antibiotics than bacteria that are not growing very well because they're kind of deeper in the biofilm. And so because they're not growing well, they won't be as susceptible to the antibiotics. So you have, instead of having one MIC in this biofilm, you'll have a range of MICs from, I can kill it to I probably can't kill it. Growing like this with this type of heterogeneity can trigger stress responses, right? So when bacteria get hungry or they can't get enough oxygen, they get stressed. And so stress responses add to their ability to tolerate antibiotics. And lastly, sometimes we get what are called persister cells forming. And these are, depending on the species, there are different mechanisms that lead to the formation of persister cells. But essentially, these are dormant cells that are highly tolerant of antibiotics. And we're still trying to understand all of the reasons behind that. But in any population of bacteria, you'll have a small fraction that become persister cells, and they become really, really hard to kill, even with very high doses of antibiotics. But they will wait around, and when you take the antibiotics away, they can regrow again. So the other thing about biofilms and antibiotic resistance is that just that biofilm environment can promote the development of resistance. So I told you that biofilms can be antibiotic tolerant, but within that tolerant population, you can get actual permanent resistance. And the reason for that is because, again, sometimes the drug concentration is too low, right? So if you just expose bacteria to sublethal concentrations, you give them time to accumulate mutations, and eventually you're going to select for a mutant that is more tolerant or resistant of the antibiotic. When you grow as a biofilm and you have all these stress pathways activated, that can induce DNA damage repair pathways that are error prone. And that means that they don't do a very good job of repairing DNA damage. And so they can create additional mutations, and some of those mutations could lead to resistance. And then, as I mentioned before, when you're growing close together like this, you can exchange resistance elements like plasmids or transposons a lot more easily because you're right beside the susceptible organism. So based on what I just told you, my lab decided to go on a search for small molecules that could inhibit biofilm formation that we could combine with antibiotics. And the idea was that we could keep the bacteria in that planktonic stage and not in the biofilm stage. And that way the antibiotics would be more likely to kill them. And, like all things in science, sometimes the question that you ask is not the answer that you find. So I'm going to tell you a story about how we went looking for small molecule inhibitors of biofilm formation and actually found biofilm stimulators with these small molecules. And that actually turned out, I think, to be almost more interesting than the original question, but I will circle back to the original question right at the end. So this is the type of assay that we use in my lab to measure biofilm formation. So this is a 96 peg lid. So it fits in a standard 96 well plate. And some people do measure biofilm development actually in the wells of a 96 well plate, because bacteria will grow, like I showed you on the rocks in the stream, the bacteria will grow around the edges of the wells in the plate. But essentially, we like this assay because you stick these little pegs into the dish, and then you get 96 very highly precise biofilms on these pegs. And so you can measure biofilm formation by 96 different kinds of bacteria, or you can test 96 different drugs at a time, or you can do dose responses. So that's what's shown in this particular image. So you can go from high to low concentrations and see how much biofilm do I get in certain concentrations of an antibiotic. So we can do this two ways. We can put the drug and the bacteria in the plate and then put the pegs in and see what happens to biofilm development in the presence of the drug. Or we can make the biofilms on the pegs first and then transfer them into a plate that has drugs in it. So that way we can test whether a drug can kill an existing or established biofilm. So in order to measure how much biofilm we have on these peg lids, we use crystal violet staining. So crystal violet is the first step of the Gram stain. You will remember, I hope. So if you dip these pegs, once they have bacteria on them, into crystal violet and then rinse it off, you can then elute the crystal violet into a separate plate using either acetic acid or ethanol. And then we measure that intensity of the purple dye on a plate reader, and that allows us to get an idea of how much biofilm was there. So we also, when we do this assay, measure the growth in the dish itself, because sometimes if you don't get a biofilm on the peg, it's because the bacteria are dead, right? And dead bacteria don't make biofilms. So what we are usually asking is, did this compound just kill the bacteria? And if the answer is yes, well, that's why we don't have a biofilm. But we're looking specifically for compounds that don't kill the bacteria, but change the amount of biofilm that we see, right? So we're more interested in compounds that can modulate the amount of biofilm rather than just kill the bacteria. So I'm going to show you some data now that sort of got us started on this path of thinking about biofilm stimulators rather than biofilm inhibitors. So we started screening using this assay, and we have at McMaster a high throughput drug screening facility, which is, full of robots, very similar to what you would see in a pharmaceutical company. And we have collections of, thousands of small molecules. But among those collections of small molecules, we have a library of about 4,000 compounds that are all previously FDA approved drugs that are no longer patented, right? And so that's the collection that we started screening with. And the organism we used originally was that Listeria Monocytogenes that I showed you the picture of at the beginning, the food pathogen. And when we do screening, we have to pick an arbitrary concentration, because you can't screen thousands and thousands of drugs at 20 different concentrations because it's impractical, right? So normally what we do is we screen our libraries at 10 micromolar. So on here, that would be, the highest concentration. The drug that I'm showing you here is ampicillin. So you may have taken this at some point for an infection. It's a pretty common beta-lactam antibiotic. And I'm going to show you a lot of these graphs that have yellow and purple. So yellow would be the growth in the dish itself, and purple is the amount of biofilm that's formed. So for Listeria, ampicillin is a really powerful drug, right? So even though we screen at 10, that was way above the minimal inhibitory concentration, right? So 0 is just our vehicle control. So it's the DMSO that the drugs are resuspended in. And we sort of set this as the baseline. So this is a normal growth and normal biofilm formation. But as soon as we tested even a small amount of ampicillin, we just killed the bacteria. So we thought we should, this might be too high as a screening concentration. So we decided to do a more broad screening concentration for this organism just to see what happened. And to our surprise, we actually, as we reduced the concentration down to this region that I have highlighted here, so under 0.63 micromolar, we actually saw more biofilm, not less biofilm. So this is the control, again, 0. But even at a tiny amount of ampicillin, right? So 20 nanomolar, we saw this spike in biofilm formation. And then as we got concentrations more and more close to the MIC, the biofilm dropped off and then we killed the bacteria. So we thought this was really interesting. And because this was on those pegs that I showed you, we actually broke off some of the pegs to look at them in the microscope. So this first image, these are scanning electron micrographs again. The first image is just the DMSO control, so 0. And this dark gray in the background is the actual plastic peg itself. And then you can see these nice little clusters of biofilm growing on it. When we looked at 0.04 micromolar ampicillin, we just saw this massive, gigantic amount of bacteria, and you can't even see the peg anymore. It's just a huge mat of bacteria. So this was like a crazy amount of biofilm. So we concluded from doing this work and some other work that if the antibiotics are below the MIC, so below the minimal inhibitory concentration that you need to kill the cells, which is about 0.3 in this for this particular drug, it triggers this, what I call an anti-antibiotic response, right? So biofilms are protecting the bacteria. So if you start treating the bacteria with a drug concentration that is not enough to kill them, their defensive response is to make some more biofilm, because that, as I showed you, has protective properties, right? So this is a way of the bacteria to quickly try to protect themselves from antibiotic before the concentration gets high enough to kill them. So I showed you data just now for Listeria. This is data from another group for methicillin-resistant Staph aureus, or MRSA, or MRSA, as some people call it. And this is just to show you that it's not only Listeria that has this response to sub-MIC antibiotics. So in this particular image, they are measuring biofilm directly in the 96-well plate, as I told you some people do. And you can see for these two MRSA strains, if you do not add any antibiotics, so just vehicle, they don't really make a lot of biofilm, right? So you're not really provoking them, so they don't do anything. But as you start to add methicillin, which these are actually resistant to, they will start to make quite a lot of biofilm until you get to a high enough concentration that you kill them. And then, of course, dead bacteria don't make any biofilm. So this phenomenon is not just specific to Listeria. It happens with lots of different bacteria. It also happens with lots of different classes of antibiotics. So I showed you ampicillin and methicillin in those last two slides. Those are both beta-lactam antibiotics, so they inhibit cell wall synthesis. But here are some data from the organism that my lab works mostly on, which is Pseudomonas aeruginosa, with fluoroquinolones, so ciprofloxacin. This inhibits DNA synthesis, cephalosporins, which is a different class of beta-lactams, and macrolides. So you can see in each of these cases, as you increase the concentration of the drug, the growth of the bacteria will eventually stop. But just before you get to that point, the bacteria start to react to the higher concentrations of drug by making more biofilm. So my students call this the, oh my God, I'm gonna die, and then they die. But we see the same pattern repeatedly. And, you know, it doesn't even need to be an antibiotic per se. So this bottom image is a 96-well plate where Pseudomonas has been exposed to soap, just very mild soap. And this, we were doing a study with some surgeons here who were trying to test this as a scrub before they do surgery. So at 100%, so undiluted, this soap will kill Pseudomonas, so you don't see any biofilm. But if you dilute this soap down, by the time you dilute it to 40% of its full strength, you can see that Pseudomonas can make biofilm in the presence of this soap, and it's quite offended by the soap because it's making a lot of biofilm. So, you know, anything that harms the bacteria will cause this response to happen. So I told you that we screened a library of 4,000 previously approved drugs, and so in that library are drugs for all kinds of things, right? So diarrhea, antipsychotic drugs, like psychiatric drugs, you name it. And interestingly, we found quite a few drugs in that collection that are not meant to be antibiotics, but they cause the same type of reaction. So they don't really kill the bacteria at these concentrations, but clearly the bacteria don't like them because they are starting to make more and more biofilm as you increase the concentration. So I have some other examples down here, but this is a really interesting thing to think about because a lot of these drugs are old and they were developed at a time when people didn't really think about your microbiome, right? So they didn't consider what effects these drugs might have on bacteria in your body. And this is not, an idea that we're the only ones who thought of this. This is a paper from a few years ago showing that if you screen that same collection of compounds that we did on human gut bacteria, there's a lot of effects on those bacteria from these non-antibiotic compounds. So, when these drugs were developed, people thought about testing the side effects on the human cells, but they didn't necessarily think about the side effects on the bacterial cells. So this is something to keep in mind if you're involved in drug development, that, just because it doesn't have an obvious bacterial target doesn't mean the bacteria don't react to it. So one of the things we're studying in my lab is how do you connect sub-MIC antibiotic exposure to more biofilm? So what is the pathway that, that's happening in the bacteria that leads to more biofilm formation? And, it's a complicated process, as you can imagine, right? Because I told you lots of different kinds of antibiotics with lots of different targets cause the same thing to happen, this increase in biofilm. So that suggested to us that it's not a target specific phenomenon, right? So it's not just that cell wall damage causes more biofilm or that DNA damage causes more biofilm, but rather that it's probably connected to stress pathways. So we published this review article a couple of years ago, and you're welcome to go read it if you're interested, where we sort of put out a few hypotheses about what is going on in the cells. So one of the things we considered is that, many bacteria contain prophages, right? And if you treat bacteria with sub-inhibitory antibiotics, you will trigger the release of prophages, because as the bacteria start to die or get very stressed, the phage will say, oh, this is a bad place to be hanging around, and they'll blow up the cell and leave. And so the release of those bacterial contents could help develop more biofilm. The second hypothesis is that I told you biofilms are physiologically heterogeneous, right? So some cells are more susceptible than others, right, within that cluster of biofilm. So one particular concentration of antibiotic could kill the more susceptible cells, but not the more resistant cells. And those dead susceptible cells will also lyse and feed the remaining cells. So both of these first two ideas kind of rely on the idea of, if you have bacteria dying and spewing out their guts, that's going to help the rest of the biofilm grow. And we tried to test that idea by, actually adding dead bacteria to the wells when we form a biofilm. And we saw a little bit more biofilm, but not anywhere near the huge spikes that we get for some antibiotics. So we don't think this idea is the only thing going on. So the third idea is that there are stress pathways being triggered, and those stress pathways lead to an increase in matrix production. And this is the hypothesis that we're working on right now, but we are not ready to show you what that looks like because we're still working on it. So in the meantime, even if we don't actually know what exactly is going on inside the bacteria, we wondered if we could exploit this phenomenon of sub-MIC antibiotics increasing biofilm to look for new antimicrobials, right? So I call this the canary in the coal mine, because the canary, miners used to use a canary in a cage to tell them if there were invisible gases that might kill them being produced in the mine, right? So we're using the bacteria as a canary to tell us if there's just a small amount of antibiotics in the test. And that way we will not miss those small amounts of antibiotics, because if we were just looking for growth, we would miss it, right? So just to reiterate that point, When we do screening, we only screen at 10 micromolar typically, right? So this is an example of Pseudomonas, Aeruginosa, a biofilm assay, and you can see for erythromycin, which is the example I'm showing you here, at 10 micromolar, if I was just to look at growth in the yellow bars, I would not consider this to be an antibiotic, right? And we all know that erythromycin is a very well-known antibiotic. But if my assay is just, did it kill Pseudomonas? I would say no, and I would have moved on. However, you can see that biofilm stimulation happens way below the actual MIC. So the MIC is somewhere, between 160 and 320. And we see this classical, oh my God, I'm going to die, and then they die, right? But you can see all this biofilm stimulation way before you get to cell death, right? So at the screening concentration, we want to be paying attention to biofilm formation, not just growth, because that way we will miss fewer compounds in our screens. So I'm going to show you an example of how this led us to find a new antibiotic activity for an old drug. So I'm showing you on the left just a subset of our screening data, right? We have hundreds and hundreds of these graphs, but this is just 4 plates of compounds. And it's just 2 replicates, right? So one on the y-axis, one on the x-axis. So anything that replicates should fall on a 45-degree line. And 1 means there's no biofilm stimulation, right? So of course, most compounds don't do anything to bacteria. And so you expect the number to be 1. But you can see for this set of plates, there are several antibiotics that stimulate biofilm formation. And of course, this is a collection that has all drugs in it. So it does have antibiotics in it. This phenomenon means to me that these are antibiotics that will kill Pseudomonas, but they're just not at a high enough concentration. So they're instead stimulating biofilm formation. So while we were going through this list, my eye was caught by this compound right here, which is Thiostrepton. So Thiostrepton is an old antibiotic, and it has, in the literature, no gram-negative activity. So Pseudomonas is a gram-negative organism. It's very difficult to get drugs into Pseudomonas. So this really caught my eye because this is the structure of this antibiotic. It's a big, huge natural product. And I thought, I don't understand why Pseudomonas is freaking out about this antibiotic, because it should never even get into Pseudomonas because it's so big. And Pseudomonas is very good at keeping really big antibiotics out of the cell. Here are the data for the biofilm assay, and you can see that there's a very obvious dose-dependent stimulation of biofilm formation when we do the dose response. And of course, at 10 micromolar, which is what we were screening, there is a strong biofilm stimulation. So this made me go, okay, maybe this drug is killing Pseudomonas at higher concentrations, but how on earth is it doing that? So this is not, as I said, a new antibiotic. It's an old antibiotic. So it's not been developed really for human use because it's not very soluble. So it's a little bit difficult to handle chemically. So it's used right now in this ointment at the top, which is a veterinary product, but it's in a mixture with other things. So you can see the list of active ingredients here. So it's only one of many things, and it's used to treat surface ulcers and things in dogs and cats. And treating things as a surface topical agent is easier when you have a very insoluble antibiotic. However, people are getting more interested in this molecule again, even though it was developed originally in the 50s, because they have recently started to discover that it has anti-malarial activity and anti-cancer activity. So people are getting interested in trying to work on this molecule, try to make it more soluble, try to make it better behaved, because it could be used as an anti-malarial or anti-cancer drug. So, these are really interesting papers to find, but it made us wonder, okay, well, so Thiostrepton has antibacterial activity, anti-cancer activity, anti-malarial activity. So maybe we shouldn't assume that what it's doing to Pseudomonas is what its known mode of action for gram-positive bacteria was, which was as a ribosome inhibitor. So the normal or the typical antibacterial activity of this molecule is to bind to the bacterial ribosome. So this is the 30S and 50S subunit of the bacterial ribosome. And this particular drug down here in blue binds in this crack between the 23S rRNA and one of the ribosomal proteins, right? So it wedges itself in here, and that messes up the function of the ribosome. So we thought, well, maybe we could test whether this, this drug is actually inhibiting Pseudomonas ribosomes. And the way we decided to do that was to try to protect the ribosomes from binding of the drug. And this particular A1067 moiety is really important for that. It's important for binding. And the bacteria that make Thiostrepton, because it's made by Streptomyces, a soil bacterium, they methylate this particular part of their ribosome to block the drug from binding, because that way they don't kill themselves, right, when they make the drug. So we took the enzyme from the Streptomyces producer, and the enzyme is called TSR for Thiostrepton resistance, and we put it into Pseudomonas. And the thinking was, if this methyltransferase can protect Pseudomonas against Thiostrepton, that would be indirect evidence that Thiostrepton was actually getting all the way into the bacteria, into the cytoplasm, binding to the ribosomes and killing Pseudomonas. And you can see that's what we saw. So this is just a killing concentration with the empty vector in Pseudomonas or the TSR expressing plasmid. And expressing TSR in Pseudomonas completely protects it from killing by Thiostrepton, which is pretty cool. So the next question became, okay, well, that looks like ribosomes are the target, but how is this gigantic molecule crossing the outer membrane of Pseudomonas to get all the way into the cytoplasm? Because, as I mentioned, it can be very difficult to get drugs into Pseudomonas because it has a very poorly permeable outer membrane. So we first considered whether it was being taken up through peptide permeases, because it's a peptide, a thiopeptide antibiotic. And we thought maybe Pseudomonas thinks this is food and it's trying to eat it and it's giving itself food poisoning. So we looked really carefully at all these transporters. We knocked a bunch of them out, and none of the knockouts had any change in susceptibility to Thiostrepton. And I think that the reason for that is that all of these drugs that exploit these peptide receptors are linear peptides, right? So these receptors evolved to take up linear peptides. And so these linear peptide antibiotics can sneak in through those receptors. But Thiostrepton is a cyclic peptide, so it doesn't fit into those peptide transporters. So while we were trying to figure out how this compound was getting into Pseudomonas, we realized through a series of experiments that are too long to tell you about today, that when we grow Pseudomonas in low iron, that will increase its susceptibility to Thiostrepton. And so that gave us a clue that maybe Thiostrepton was sneaking in through iron transporters rather than peptide transporters. So Pseudomonas makes this molecule called pyoverdin, which I'm showing you the structure of here. And that's what gives it this crazy green color when you grow it on media that has low iron. So it sends this molecule out. And when the molecule binds iron, it quenches that fluorescence. And the bacteria recognize the iron-bound form and they bring it back into the cell. And so we decided to test whether Thiostrepton was using the receptor for this molecule. Because we saw that if you add an iron chelator, right? So if you chelate iron using this molecule called EDDHA, you increase the susceptibility of Pseudomonas to Thiostrepton, right? So you actually shift the biofilm stimulation to lower concentrations, because now in low iron, they would make more of these receptors trying to get more iron. If you add iron, you actually shift the susceptibility the other way. So if you give the bacteria iron, they dialed down the production of receptors for pyovaridin, and so less of the antibiotic would be getting in. And so the receptor for pyovaridin, there's actually two in Pseudomonas. One's called FPVA, so pyovaridin A, and the other one's FPVB. And we knocked out each of these individually and then both of them together. And only when we knocked out both of the pyovaridin receptors together did we get resistance to Thiostrepton, which is shown in this light gray bar. So these are called TUN-B dependent receptors because basically they are an outer membrane pore with a plug in it, and you need energy coming from the proton motive force of the inner membrane through this TUN-B complex to pull the plug down when the ligand binds in the top. So Pseudomonas also makes a separate receptor for a molecule called pyocheilin, but when we knocked that out, it didn't do anything to susceptibility. So we realized, okay, that Thiostrepton is hijacking these receptors. And we wondered, well, is it doing that because it binds iron and the bacteria think that they're getting an iron-bound molecule, or does it just look like pyovaridin bound to iron? So we did this little quick assay, which shows you whether or not a molecule might bind iron. This is chrome azaryl S agar. It has a blue dye in it that binds iron. And if you add an iron chelator that is stronger than the dye, and it will steal the iron from the dye, then you'll get a yellow or orangey color, right? So these are two control iron binding molecules, deferoserox and deferoprone. And both of them can strip the iron away from the dye, but Thiostrepton could not do that. And neither could the control vancomycin. So we don't think Thiostrepton actually binds iron to exploit these receptors. I think that it has just evolved to be in the shape of pyovaridin bound to iron. And so it fools the bacteria into taking it up. So because we realized that this thing is iron dependent, right? So the lower the iron in the media, the more the bacteria make the receptors, right? They make more receptors and they try to bring in more iron because they're starving for iron. If you combine Thiostrepton with FDA-approved iron chelators, you can actually sensitize the bacteria even more to Thiostrepton. So these pictures that I'm showing you are called checkerboard assays, and they have two different drugs on the two different axes, right? So Thiostrepton is on the y-axis, and deferoprone or deferoserox, which are iron chelators, are on the x-axis. So individually, they don't do a ton to the bacteria. So deferoprone will stop Pseudomonas from growing at very high concentrations, but at lower concentrations doesn't do anything. And Thiostrepton is not a great drug on its own. But when you combine them, you can get this classic staircase pattern, which shows synergy, right? So that means that you can use a lower concentration of Thiostrepton if you add the iron chelator, and you can still kill the bacteria. Deferoserox works even better. It's fantastic. We also tested the concentration of Thiostrepton that you would need to kill Pseudomonas in serum. So serum is already naturally low in iron because your body makes iron binding proteins to keep bacteria from growing. And you can see whether we used mouse serum or human serum, it worked fabulously well, right? So serum alone does not, or serum alone with the chelator does nothing, but serum with thiopeptides looks fabulous, right? So this was a great discovery, and it also helps in terms of the fact that this drug is not very soluble, right? Because if you don't have a very soluble drug, but you don't need to use nearly as much of it, that's great, right? So then you don't have to worry so much about solubility. So this discovery made Derek, my student who's working on this project, wonder whether other thiopeptides or even other natural product antibiotics have actually evolved to exploit this class of receptors for their uptake. So he showed in work that I don't have time to show you today that another thiopeptide called thiocillin also exploits iron uptake receptors, but not the pyovaridin ones. Instead, it uses this receptor called FOXA, which is for a siderophore called ferrioxamine. And the interesting thing about this is that Pseudomonas doesn't even make ferrioxamine, right? So if it doesn't make ferrioxamine, why is it making this ferrioxamine receptor? Well, it turns out that Pseudomonas makes two of its own iron binding molecules, which I mentioned, so pyoveridin and pyochelin, but it also has the capacity to steal other bacteria siderophores, right? So remember I mentioned at the beginning about microbial cheaters and how you could have organisms that steal other bacteria's goods and they don't actually make them themselves. Well, Pseudomonas is really good at that. So it makes a lot of these receptors to steal things from other bacteria. And so one of those receptors is FOXA. It's meant to steal ferrioxamine, but it also allows thiocillin in. So Derek decided to expand this idea and developed what he calls the SURFR platform, SURFR for surface receptor, right? So we looked in the genome of Pseudomonas and we found 34 different predicted TUMB receptors. And so he cloned each one of those individually into an arabinose-inducible vector. One of them was toxic when he tried to clone it, so we couldn't use that one. And 3 of them, when you overexpress them, can make the bacteria sick. So we keep the arabinose concentration low. But the idea was if you take this set of strains now and try treating them with a whole bunch of different natural products that normally would not kill Pseudomonas, can you find the receptors for these drugs by doing this? So here is across the top, the numbers don't really matter. What these are the gene names from Pseudomonas, but each of them is an individual receptor overexpressed in the wild type background. And the first row or column rather is the empty vector. So P herd is the vector that we use. So if you have just the vector in Pseudomonas, of course, the bacteria can grow. But if you have either FPV-A or FPV-B overexpressed in the wild type, you will kill the bacteria if you treat them with Thiostrepton, because now you've made extra receptors and this drug can get in. You do not see that for FOXA and Thiostrepton, but for thiocillin, which is the one I just mentioned, you see killing when you overexpress FOXA, but also this receptor, which is uncharacterized. So that suggests that, like Thiostrepton, thiocillin has 2 different things that it can exploit to get into the cell, which is kind of cool. So Derek has just started doing these screens, but I wanted to show you one example of what he's found. So here is a whole bunch of different antibiotics down the side, and you'll see here vancomycin. So vancomycin is a large antibiotic. I showed you the structure at the bottom, and this is not used to treat Pseudomonas because, again, it does not get into the cells, right? And you can see that if you look at the control lane, right? Pseudomonas with just the vector, it's growing just fine in the presence of vancomycin. But Derek found that one of these uncharacterized receptors actually allows vancomycin to kill Pseudomonas. So, you know, this is just the beginning of this project, but we think this is pretty cool. And it may also explain why these molecules are so big and crazy looking, right? Because maybe they have evolved part of the molecule to bind to their target, which is the ribosome, but the other stuff on the molecule has evolved because it can bind to the receptor. Right? So you need both of those kind of warheads to get the molecule into the bacteria and then onto its target. Okay, so I'm just going to end my talk by bringing you back to our original goal, which was to inhibit biofilm formation with small molecules so that we could try to help antibiotics be more effective. So while we were doing this screening, we found some biofilm inhibitors and we started looking more carefully at whether they were able to chelate iron, because we were interested in iron chelation for combining with the thiopeptides because we want to keep the iron concentration low for reasons I just explained. But we were also aware of this paper, which is from 2002, which showed that if you chelate iron, Pseudomonas has a hard time making a biofilm. And in this particular paper, they used lactoferrin, which is one of the proteins that your body uses to chelate iron. And you can see that in the absence of lactoferrin, Pseudomonas makes very nice biofilms. And these are confocal micrographs of these big macrocolonies that I showed you at the beginning. But if you add lactoferrin, the bacteria do not make very good biofilms, right? And they tied this to the fact that the bacteria increase their motility in the absence of iron because they don't like to stay in an environment where there isn't very much iron. So they try to increase their motility and leave. So we went back and looked at, hey, if we have a biofilm inhibitor that looks like it chelates iron, maybe it will do a dual purpose, right? It will reduce biofilm by chelating iron, and it will increase susceptibility to thiopeptides. So we could have both of the things that we want at the same time. So these are just examples of 6 compounds that came out of our recent screen for things that inhibit biofilm. So in the purple and yellow graph, you can see that these compounds don't inhibit growth, but they also block biofilm formation to various extents. And in the blue and pink graphs, we have the effect of the compound alone on growth and the effect of the compound when you combine it with Thiostrepton. And you can see that many of these compounds will kill the bacteria in the presence of Thiostrepton will kill the bacteria in the presence of the compound because it's chelating iron. And this one I really like. This is the one that we're working on the most because it's really potent at potentiating Thiostrepton activity. And it's also a decent biofilm inhibitor. So I just want to point out that these are log scale graphs. So this is actually pretty good inhibitory activity. So we did actually get all the way back to finding biofilm inhibitors at the end of this project. We just took a very kind of circuitous route to getting there. So just to summarize what I told you today, biofilms are complex surface-associated communities, and they protect bacteria from threats, including antibiotics. I showed you that sub-inhibitory antibiotics can stimulate biofilm formation and that you can use this phenomenon as a tool to find new drugs. And I also gave you a story about how thiopeptide antibiotics, which we discovered through this biofilm phenotype, exploit nutrient receptors, specifically iron uptake receptors, in gram-negative pathogens. And this is giving us potentially an idea of how we could find ways to get really large antibiotics into difficult to treat bacteria so that we can expand our repertoire of antibiotics for these difficult pathogens. So I just want to say thanks to all the students that work on these projects in my lab. So Derek in particular is working on the thiopeptide project that I just showed you and our funders, and I would be happy to take any questions.
All right, so thank you very much, Professor. It was a really, really engaging lecture. I personally was very, very taken by every word that she said, and I hope that our audience liked it too. We're ready to take questions. I also have some of my own and also some from the chat. So if someone wants to turn on their microphone, they're welcome to do it now. All right, then I'll read the question from the chat. Someone is interested in knowing if this kind of research, what in general can we expect in the future? How can it be used in order to prevent the spreading of resistance? And maybe if we know that some antibiotics that previously were not used for treating a pool of bacteria, can we use them now if new types of functions are found, as was the case of Thiostrepton?
Yeah, so great questions. So first of all, just knowing that sub-inhibitory antibiotics will trigger increased biofilm formation is a really important thing to know about, right? Because I showed you that lots of compounds can do this. And so, you think about how you're going to treat an infection, you want to be aware that this could be a problem, right? So one of the places I could see this is important is, for example, in cystic fibrosis patients, right? So they have lung infections with Pseudomonas that are chronic, and they're so difficult to treat that they get inhaled antibiotics, right? And so you can imagine that when you inhale an antibiotic, you get a high local concentration at the top of your lungs, but as it has trouble penetrating down, you may actually get to the point where the concentration has dropped below the minimal inhibitory concentration for that bacterium. And you could actually make the problem worse by stimulating biofilm formation in the lung, right? So it's important to know that this is happening. It's also important to know how it happens. And that's one of the things, like I said, that we are trying to study, because if we understand the pathways that lead to more biofilm, we can try to disrupt those pathways, right? So one of my PhD students, Luke, is looking for mutants of Pseudomonas that cannot make more biofilm when you treat them with sub-MIC antibiotics, right? So he's found a few mutants, and not very many, because our screen for the mutants is that they have to not respond to 3 different classes of antibiotics. So that's a pretty high demand ask. But we have found some stress pathway genes that when you knock them out, the bacteria seem to be blind to the antibiotic. So we are pursuing those as potential leads for things that we can disrupt. And then the third part of the question was, how can we use this to find new drugs? Well, this is exactly what we're doing right now. So in addition to the thiopeptide story that I just told you, and about Derek's Surfer platform that he's using to look for other natural products that can exploit these pathways to get in, we have in our screens, I showed you like one snapshot of the data. We have many more compounds that we identified that stimulate biofilm formation. And that tells us that they are probably antibiotics, but just not antibiotics at 10 micromolar. So we have taken a couple of those compounds and done medicinal chemistry on them to make them more potent, right? And to make them better antibiotics. So those stories are coming. You have to stay tuned.
We hope they are. And another question of mine, I'm interested in the how. So you mentioned basically 3 hypotheses that we could put in 2 classes. One that's related to dead bacteria initiating biofilms, and the completely different approach related to that. So could there be maybe a positive interference between those, so that some stress pathways could be triggered by the population of bacteria dropping as a consequence of antibiotic treatment, or something in that sense?
Absolutely. So as I said, one of the things we tried was, just adding, because one of the early matrix components is DNA. So we tried just adding DNA to the bacteria as they're forming a biofilm, or adding sonicated bacteria, or adding autoclave bacteria, like whatever we could think of to try to mimic dead bacteria being in that well. And the biggest difference we got was maybe 200% of our control. But sometimes when we treat with a sub-MIC antibiotic, I showed you for erythromycin, right? We get ridiculous stimulation, right? So it cannot be explained simply by dead bacteria, enhancing biofilm formation by the rest of the population. So Luke's research right now is pointing the genes that are knocked out in the mutants that don't react are all related to central metabolism, surprisingly. And so the current hypothesis that he's working on is that when you treat the bacteria with a little bit of antibiotic, you do some damage to the bacteria, right? But they don't die. So in order to try to fix the damage, they need to increase their ATP levels, right? Because now they have to do all these processes to repair things that have been busted by the antibiotic. And so the processes that he's seeing that are knocked out in these mutants have to do with ramping up how much ATP you can make, right? And so if you prevent them from doing that, then they don't seem to be able to increase their biofilm.
Thank you very much. And one more question, if you can, please. I saw on the little scheme where Thiostrepton binds the ribosome, there are some proline residues involved in the process. And proline is my favorite amino acid. So I would be very curious in finding out whether these prolines, what happens if they're mutated, or are they what kind of interactions are formed between the binding structure, so between the Thiostrepton and those prolines?
Right. So that is a fantastic question and very perceptive. The producers of Thiostrepton and other thiopeptides make, so they have, your normal ribosomal protein complement, but many of them make a separate copy of L11 in the cluster that makes the Thiostrepton antibiotic or the thiopeptide antibiotic. And that version of L11 doesn't have the same amino acid sequence. It's missing the proline. So in that case, when they start to make, they express the biosynthetic genes to make the drug, they also express this alternate form of this ribosomal protein. So at the same time they make the drug, they protect themselves by making this other L11 protein. And we have actually, for thiocillin, I didn't talk about that work because I didn't have time, but for thiocillin, we didn't have an equivalent protection protein like TSR that we did for Thiostrepton. So instead, Derek changed the sequence of the Pseudomonas L11 protein to make it look like the producer protein, and that also protects the bacteria. So that protects Pseudomonas from thiocillin activity.
It's amazing. I'm so happy now for that.
I know, bacteria are so cool, right?
Of course, and proline, especially proline in bacteria. It's best to have proline in bacteria. All right, if no one else has any more questions, please, if someone has, do speak up. All right, it seems that's it for the evening. I'd like to thank you very much, Professor, for this lecture and also for the Q&A session. It was really interesting and engaging. So I'd like to thank you and also to thank our audience for joining us this evening in Serbia during the Antimicrobial Awareness Week. Stay tuned for the next part of our program and the lecture, which is due on Sunday. Someone is saying thank you very much. You're very welcome. Thank you very much again, everyone. And until next time, help us and spread awareness to stop resistance. Thank you. Goodbye.
Thank you.